What is going on? It is that time again, Wednesday, March 2nd. February is done and gone. I missed my initial March video, so I know all of you who are watching are so sad. It's okay. My March 1st video, or my March 1st learnings will somewhat be encompassed into today's learnings. But yeah, just to remind everybody, the last place I left off was going more into cellular structures, and I was talking about the nucleus, the nucleolus, all the different, on a higher level, all the different organelles which kind of exist in the cell and which are anchored through the cell in various ways via intermediate filaments. But yeah, and I'm going a little, maybe haphazardly, jumping around a little bit, but I think it's important just to kind of really anchor our understanding and knowledge of the cell structure and cell division and everything like that, so that when we go into the deeper elements of cell growth and DNA and whatnot, we can refer back to this knowledge of cellular structure to kind of have a better understanding of how things get done at the genetic level, because all of the processes and all of the synthesis of enzymes like DNA polymerase, etc., all of that is done through the cell's cycle, various cycles, various like intermediate processes. There's so many things happening within a cell that, it's biology is really sort of the art and science of connecting things together, right, in order to achieve real understanding. So that's what we're kind of doing here, building that foundation and going back to the foundation and re-cementing it, etc. So enough of explanation of my strategy here in how we're learning. So we will start picking up with the rough endoplasmic reticulum and smooth endoplasmic reticulum. So firstly, we know what the rough endoplasmic reticulum looks like. This is probably a good example, right? Your rough endoplasmic reticulum is this part right outside the nucleus of the cell, and it contains ribosomes, and it's involved in protein synthesis and protein modification through glycosylation. Okay, so we will also talk about, there's two other things we should talk about, which is the smooth endoplasmic reticulum and the Golgi apparatus. So once proteins are synthesized, they're packaged into vesicles and bud off from the rough endoplasmic reticulum and get sent over to the Golgi apparatus, where they are sent over to the lysosome, to the cell wall to maintain cell structure, and to bud outside of the cell. So basically, the Golgi is the Amazon of the cell, right? Amazon delivery system of the cell. But the smooth endoplasmic reticulum, look, so if proteins are synthesized in the rough endoplasmic reticulum and folded there, the ribosomes that are bound to the rough endoplasmic reticulum are responsible for the creation of those proteins, these structural proteins and other very specific types of proteins. Then the smooth endoplasmic reticulum is responsible for other molecules, for example, fatty acid synthesis, right? So, and gluconeogenesis and other processes that involve sugars and fats. So, for example, fats are created, and the smooth endoplasmic reticulum also looks different, right? I think it looks more like this organ-like structure, as most people in school like to say, or this coral-like structure. And so fats are created and synthesized through various processes in here. It's all, the smooth endoplasmic reticulum is also responsible for calcium storage. There, in muscle cells, for instance, there's a analogous structure called a sarcoplasmic reticulum, which is really good at calcium storage, because the muscles need calcium to activate the neurotransmitters, right? Like we went over that a while ago, or I'd gone over that a while ago in a different video. But yeah, so muscle contraction, etc., that's essentially facilitated through smooth ER, but I think all cells have some sort of calcium storage in their smooth ER. But the smooth ER also is responsible for creation of enzymes which degrade alcohol and ethanol and other hydrogen peroxide. Okay, so that it's basically responsible. It's in your liver. I think the enzyme is CD45 or something. There's a specific type of enzyme which I will research, which is responsible for all that, which is created by the smooth endoplasmic reticulum. So basically, the same process occurs for the smooth versus the rough in terms of the budding off of these fatty acids and sugars and other things that occur that are created that are not proteins, and they are sent to the Golgi apparatus through a process which I've been curious about. Like, how does the bud know to go to the Golgi apparatus, right? And there must be some sort of signaling or some sort of chemotactic process going on, which is a process that would lead the bud through chemicals, chemical markers to the Golgi apparatus, but that's something to think about later. But at the Golgi apparatus, so Golgi apparatus or Golgi body is another structure, which it's another commonly known structure, but it's very important, and it looks like kind of this, I don't know, pancake thing, right? So the Golgi apparatus is responsible for many things, mostly packaging, as you know, but its structure is that it has a cis phase and a trans phase. And the cis phase is where the molecules come in, they're incoming transport vesicles. The trans phase is where the molecules leave in secretory vesicles and go to the cell wall, or they go to other organelles or wherever they need to go to. And the different pancake structures themselves of the Golgi apparatus are called the cisternae, the interspace of the Golgi apparatus. In the same way, the interspace of the endoplasmic reticulum is named the lumen. Okay, and so through each of these pancake cisternae structures in the Golgi apparatus, protein modifications several things can happen, but one of them is protein modifications that were happening in the rough endoplasmic reticulum can also happen in the Golgi apparatus. But the protein modifications, remember I talked about glycosylation. There are two types. There's N-glycosylation of serine proteins, and then there was O-glycosylation of threonine threonine amino acids. So basically, remember what glycosylation is quickly. It's the addition of a sugar molecule, glycosylation, adding of glycose to a protein by addition of an amino acid linking covalently to one of the side chains of the protein, right? And so usually the side chain is linked either through the nitrogen bond of the amino acid or the oxygen bond, which is the carboxylic oxygen bond or carbonyl oxygen. So basically, this one thing to remember is that the Golgi apparatus can perform both of these types of glycosylation, N-glycosylation and O-glycosylation. The other structures, like the ribosome, can only perform N-glycosylation. So that's important to remember because that differentiates the modification function of the Golgi apparatus, right? So this is kind of like a little punch card kind of function. Modified molecules are secreted out of a cell to another organelle. So that's what I was just talking about, the modifications of the proteins that get packaged through the Golgi apparatus. So that's another thing to talk about. And then next, now I'm probably going a little bit fast, but I did want to talk about this. And this is quite a complex structure, the lysosome. And the lysosome is something we want to definitely, when we're talking about our eternal life studies, get super deep into. I think this is probably a good diagram. Right, so the lysosome is a fundamental organelle within your cell, which is responsible. It contains a bunch of different enzymes, And these enzymes are all grouped via different functions. So some are nucleases, others are lipases, others are proteases, and others are amylases and other kinds of sugar degradation proteins. And as the name indicates, the proteins, the digestive proteins, are focused on a specific kind of macromolecule, right? So lipases are for fats, the proteases are for proteins, nucleases are for nucleic acids and the breakdown of viruses, viral DNA and RNA, and the amylases are for breakdown of sugar. So the lysosome is multifunctional, and it's activated through several processes. It also interacts itself through several processes, and we can go into that in its whole on separate video, but briefly, it's needed for destruction, right? Like, so it's garbage collects stuff. That's the common parlance. Things are brought to the lysosome, and they're digested. When they are brought in to the lysosome's barrier, the lysosome can also itself consume things through the interaction of something called a phagophore. That's another whole other area, and these are all categories of what's called cellular autophagy, or self-eating. When the lysosome is responsible for eating eatable components of the cell, which could be like cellular garbage or other things. And then the final type of autophagy, this large-scale autophagy that occurs, is when the lysosome bursts, right? The lysosome will, if the cell needs to die for whatever reason, it's tagged for apoptosis because the cell is too damaged to replicate, or whatever reason. The body will, sick cells can signal other cells to undergo apoptosis. The primary controlled way of doing that is not cellular lysis, or not membrane lysis of the cell through exploding the diffusive pressure, but it's rather the breakdown of lysosomes, which live within the cell, which are anchored again by intermediate filaments, like all the organelles. The lysosomes break down, and they release the digestive enzymes into the cell, which all the different kinds of digestive enzymes act on all the different components in the cell, and basically the cell's inner particulate matter is dispersed after the cell dies, and it's consumed by white blood cells and other things that try to clean up your body from dead cells. So, or they left your body. So that's basically what the lysosome is important for. We need to get into autophagy on its own, in its own merits, because it's important for life extension, as I mentioned before. And so the next thing we'll talk about is the mitochondria, but I've already gotten pretty far into this video, and I don't want to go through all the different pieces of the cell. Plus the mitochondria is pretty well known and exceptionally complicated, like the other organelles, so it's worth having its own little venture into. Okay, so that's it for today. It's your bio day. Have a good day. I'll see you around, and stay bio. Okay.